Hello, everybody. I am Dr. Raktim and I am here to discuss on the topic of investigation of an outbreak. Let us begin. What is an outbreak? An outbreak is the occurrence of cases or illness, health related states or events in a defined community or region clearly in excess of normal expectancy. Here, we have to remember one thing that outbreak occurs in defined community. It is a smaller geographical area. Similarly, you may come across the term epidemic. Epidemic is same as outbreak, but epidemic usually covers a greater or larger geographical area compared to an outbreak. There also, the number of cases should be clearly in excess of normal expectancy. With clearly in excess of normal expectancy, what I mean is that in this graph, you can see that on 22nd of March 2021, there were 5 cases at this point. And when the number of cases of a disease have increased, okay, so the usual number of cases that a community has for a particular disease is the term known as endemic, while the outbreak or epidemic, it has more number of cases. With more number or excess number of cases, what I mean, we will be discussing it in subsequent slides. But for now, it is clearly in excess of normal expectancy, means the number of cases with which it raises a concern, starting with 5 cases here and going up to 16 cases here, there is a sharp increase. So we can term it as outbreak. Looking forward to the magnitude of an outbreak, on what factors it depends upon. It depends upon the agent, type, and population exposed. When a disease have a very potent or virulent agent, then the outbreak may happen. The type of outbreak. The type of outbreak is whether that outbreak has occurred from a single common source with multiple exposures or single exposures, whether the transmission is common in person to person, or when the population exposed have got immunity towards it or not. These are important factors on which the magnitude of an outbreak depends. It also occurs. Uh, it also depends upon the time and place of occurrence. The time and place of occurrence is whether uh, it is in a high populated area or a minimum populated area where the number of susceptible individuals are high or low. It also depends upon those factors. Thirdly, the previous exposure to the disease. In these 3 bullet points, I can cite you one example of COVID 19, which started in Wuhan, China. The agent was COVID 19 virus, which is very virulent, and the type was propagated in nature, where the person to person transmission was high, and the population exposed was not at all immune to COVID 19. The time and place of occurrence, it occurred in a market, started in a Wuhan market, where it was highly populated, with a population where immunity was not there for COVID, and they had no previous exposure to the disease as such, and thus it led to the dreaded pandemic. Okay? So these are the factors on which the magnitude of an outbreak depends. Moving forward to the types of outbreaks now. Just like I said, it can be from a common source, and the common source, it may be from a single exposure or a multiple exposure. When it is single exposure, it is known as point source, starting from a single exposure in a single point of time. Multiple exposure means the exposure to the disease agent is present in the environment, and the susceptible population are exposed to that uh, disease causing agent for multiple number of times. So it is also known as continuous source. Thirdly, the propagated epidemics. Propagated epidemics, I mean, when the disease is transmitting from person to person, arthropod vector, animal reservoir. These are few schematic diagrams of different types of outbreaks, where this is the point source. We starting from a single case here and going up. In continuous, there is a constant presence of a disease. In propagated, starting slowly and then it is going up. Okay, and intermittent is just like there is outbreak intermittently. This is the outbreak time. These are the outbreak times, and in between, this is the disease-free interval. Okay, we will be discussing it in subsequent slides. So I'm looking forward to the common source epidemic now. In common source epidemic, it can be single exposure epidemic. Number one. So in single exposure epidemic, it is also known as point source, starting from a single point, just like this diagram. It starts here, okay? And the number of cases gradually increases. But once the cases increases, there is a steep rise of cases, and then it reaches a peak. This is the peak, and then there is a steep fall. There is a steep rise of cases, reaches a peak, and there is steep fall. That is, with this we know that the disease agent, the exposure is brief and simultaneous, and all the cases develop in one incubation period. As I said, there is rise and fall rapidly. So when will you see this kind of epidemic? It is generally seen uh, in case of food poisoning, where suppose you are visiting a wedding party and then you are having food from the party. All the persons who are who went there to attend the wedding, and the source of epidemic for that may be the food that was served in the wedding, and there all the persons are susceptible. From there, that was the single point source, single exposure. It was brief, and all the resultant cases will develop in one incubation period. In case of food poisoning, there will be steep rise of cases and then it falls off rapidly. That was all about the single exposure. Now coming to the common source epidemic of multiple exposure, or it is also known as repeated exposure epidemic. So in this case, what happens? Since it is continuous in spread, so here there will be continuous presence of the susceptible population and the cases, since the source is common and it is repetitive. It not necessarily be at the same time or place. Mark this. It not necessarily be at the same time or place. The epidemics are extended or irregular. It lasts for a longer period of time, and the number of cases ranges from more than one incubation period. Just like you can imagine the clients of female sex worker, that female sex worker is a common source, and the clients to that female sex worker becomes the susceptible mm, population for sexually transmitted diseases, and it will occur continuously. This epidemic will tend to remain for a longer duration of time. Similarly, suppose in a community well, the community well is contaminated, and that well water is the common source to all the population out there. And since they are drawing water from that well and using it, all the community members would be mm, susceptible for a waterborne disease. And that epidemic is a continuous exposure, common source epidemic. Now coming to the propagated epidemic. In In propagated epidemic, it is mostly infectious in origin. It generally does not occur in non-infectious diseases. It is seen in infectious diseases. So there is gradual rise and tails off over a much longer period of time. So this is the diagrammatic figure. Here at this point there was only one case. With time, it gradually development of cases have begun. In this point there are four cases, and then again there is a brief interval where there are no cases, and then it rises, reaches a peak. This is the peak level, and it continues to be present. In this point there are cases, so there is gradual rise of cases, and it tails off over a much longer period of time. These kind of epidemics are generally seen in case of infectious diseases, and the transmission continues till all the susceptible individuals are no longer exposed to the infected person. So how to control these propagated epidemics? It depends upon the herd immunity. Number one, why I am saying herd immunity? Because imagine that in a population there are susceptible individuals for a disease, just like measles. Measles is infectious in origin, and the susceptible population are there for measles. So if you give vaccination, measles vaccination to the population, then that particular individual will be uh, will not be susceptible for measles. So the case of measles, to spread the disease to the other, that case of measles will require susceptible population nearby. And if the nearby population to that measles case is no longer there, then that case of measles will not be able to transmit measles infection to the susceptible. So, if you make a barrier near the case of a particular disease so that no susceptible individuals are present, it is termed as herd immunity. When you will create a barrier among near the case of a particular disease, then there will be very less opportunities for contact with the susceptible population, as a result of which the number of cases will decrease. On the other hand, if the herd immunity is not present, and if the opportunities for contact is more, then the transmission of the infectious disease would be more, and it will lead to higher magnitude of the propagated epidemic. Thirdly, it also depends upon the secondary attack rate. With secondary attack rate, what I mean? Suppose a person is infected with an infectious disease, say measles. Then the number of cases of measles, the susceptible individuals may contract after coming in contact with the uh, primary case of measles. That is known as secondary attack rate. So it also depends upon the secondary attack rate. When the secondary attack rate for a particular disease is high, then the chance of disease transmission is more. As a result, the chance of propagated epidemics is more. On the other hand, if the secondary attack rate is very low, then the chances of transmission is low. As a result, the propagated epidemics will not occur. Moving forward to the objectives of investigation of an outbreak. So we will be discussing about the objectives now. It is very simple. Firstly, to define the magnitude of the outbreak. Suppose you have confirmed that there is an outbreak going on in the community. The first thing, what will you think of? You will think of uh, the persons, how many persons are affected with outbreak. So that will determine the magnitude of the outbreak. You will be thinking about the age and sex-wise distribution, about the mm, cases with relation to time, place, person, isn't it? So that will all determine the magnitude of the outbreak. Secondly, you will think of the factors responsible for the outbreaks. What led to these outbreaks? That is the obvious question that comes to our mind. Just like I gave the example of food poisoning, so you would be thinking that how many persons are affected with this food poisoning outbreak, and what are the factors, which food were responsible for this outbreak. So definitely, to define the magnitude of the outbreak. Secondly, to determine the factors responsible for outbreak. Thirdly, to identify the source and mode of transmission. So we will also think about the potential source. Just like in case of food poisoning, the source is the food in a community well, or any just like in case of COVID, it was Wuhan market, that was the source. And then you will obviously think of the mode of transmission of the outbreak. So with these three, you may know that what kind of disease are you dealing with. So these are our objectives. The fourth objectives. With all this knowledge, you will determine the necessary measures to control the outbreak. This was very much evident in case of COVID-19, as you might notice that the government declared that hand washing, wearing of masks, and this social distancing was very necessary to control the outbreak. With all these factors of defining the magnitude, determining the factors responsible for it, and the source, they declared that COVID-19, to control it, we have to have hand washing done properly. Social distancing is a must. Okay, so these were the necessary control measures immediately taken. Similarly, for other diseases, the necessary measures will have to be taken to control the outbreaks. And fourthly, the recommendations to prevent its recurrence. The government says that for in case of COVID-19, to prevent its recurrence, we have to follow all this social distancing, hand washing, hmm, unnecessary contact with other persons should be avoided. All such things were recommended to prevent its recurrence. So to investigate an outbreak, the objectives, these are the objectives for it. Again, I repeat, to determine the magnitude of the outbreak, factors responsible for the outbreak, the source and mode of transmission, the necessary measures, and recommendations. These are the objectives. Now, since we know about the objectives of investigation of an outbreak, let us now see the steps of investigation of an outbreak. But before that, let me tell you frankly that here uh, I will be discussing the 10 steps of investigation of an outbreak. There are, in fact, 10 steps, one step by other. But in real world, what happens that there is no clear-cut definition. The first few steps are not done in a real world because the epidemiologists are called at a very later stage. When outbreak is suspected, the epidemiologist should be immediately called upon. On the other hand, uh, since there is a delay, the first few steps are often missed by epidemiologists. But for the sake of our understanding, we have to know the steps of investigation of an outbreak. Let us discuss one by one. Number one, verification of diagnosis. Verification. So before someone calls this to be an outbreak, we have to verify. This is our first step in investigation. It also comes as a short note, MCQ, and a Viva question. So do remember that the first step in investigation of an outbreak is the verification of the diagnosis. Because sometimes what happens that the reports or the news may be false, or due to any misinterpreted data, someone will tend to say that this is an outbreak. So we must verify that whether this disease or diagnosis that is occurring is an outbreak or not. Secondly, after verifying, we have to confirm the existence of outbreak. With this, I mean the diagnosis should be confirmed, number one, with the case definitions. Secondly, we have to confirm whether there is an epidemic going on or not, or whether it is an endemic. So these are the two most important steps of investigation of an outbreak. So after this, what happens? After verifying and confirmation, we will say that this is an outbreak. Okay, these are the two steps. And how the confirmation is done? We would be comparing the disease frequencies of that particular disease according to the case definition, and then comparing it with the previous year's data. And it is said that arbitrarily the limit of two standard errors is considered to be an epidemic. Just like in a study sample, we calculate standard deviation, that is a measure of dispersion. Similarly, standard error is standard deviation in the population level. So when there is deviation of more than two standard errors, then we will say to be an epidemic. Just for example, if the usual occurrence of a disease in a community is, let's say, 4, okay, then more than 2 standard deviation means it is more than 8. Okay, so if the number of cases of that disease in that particular community turns out to be more than 8, then we will say that an outbreak of that particular disease has occurred in that community. It means that. Now, moving forward to the third step. Okay, the third step is we have to define the population at risk. It is very important. Firstly, we have to verify the diagnosis, whether it is correct or is it misinterpreted. Secondly, we have to confirm the presence of an outbreak, whether outbreak has really occurred or not. And thirdly, we have to define the population at risk. And how will you do? How will you do the defining a population at risk? We have to obtain the map of the area. What area is affected? Who are the population living there? What is the age and sex-wise distribution of the population that are affected or are at risk? So we need to obtain the map of the area. Here, the mapping is done by house-to-house visit, and then a map is created where we will give the landmarks of that particular area where the population at risk is present. We will be marking the streets, okay, the church, temples, shop. All these things will be done, and then we have to map the area. And with mapping the area, we will be counting the population, how much population that area caters, so that we know the denominator, that out of how much population the disease have occurred uh, among whom. So the counting of population is done by house to house visit. Similarly, during the house to house visit, the map of that particular area is drawn. Next, after doing the mapping of the area and counting of the population, we will do the rapid search of all cases. This third and fourth point are done simultaneously. When a health worker visits that particular area and he or she search for the population at risk, that person would be drawing the map, that is also known as mapping of the area, counting the population, how much population is there in that particular area. And as in addition to that, the person will be also responsible for rapid search of the cases. Firstly, the rapid search of cases is done by medical survey, looking into the case definition, the clinical sign and symptoms, the distribution by time, place, person. Okay, the investigations done with all such, uh, we term it as medical survey, and with medical survey, rapid search of cases is done. Secondly, the epidemiological cases. When with the medical survey, the uh, population at risk or the probable case of that particular disease is identified, then a detailed history is taken in the form of epidemiological case sheet. A thorough history with the clinical patterns, how and what are the factors responsible for the sign and symptoms, and who else are involved or who else are affected with the same kind of sign and symptoms, who are the uh, contacts that have come with the probable case, the contact tracing. All such things are noted down in the epidemiological case sheet. And with that, with the same case definition, we would be searching for more cases, more similar cases, or the persons having the most similar sign and symptoms or case definition by house to house visits. So, in this way, the rapid search of the cases in that community or population is being done. I have given this logo. This logo is of Epidemic Intelligence Services. This logo, where the persons in the Epidemic Intelligence Services tries to do all these steps of investigation of an outbreak. It is also comes under shoe leather epidemiology because the persons here are visiting house to house in a community and asking for all this information. And since due to visit to every house and gathering these informations, their shoe might have a hole. So this is the logo actually, which is taken up in epidemic intelligence services. And I have put this logo because I wanted to sensitize you all with the term epidemic intelligence services, EIS, and shoe leather epidemiology. The shoe leather epidemiology comes under this investigation of an outbreak. Moving forward to the next step now, the next step is data analysis. After gathering all the informations from the community, we would have a large number of data. These data are analyzed systematically, and then definite graphs and charts are being formulated. This data analysis mainly is focused on the time, place, and person distribution. Time, place, and person distribution because we have to know that how age and sex-wise distribution of the population, who are the population at risk, what is the source of this outbreak, what is the mode of transmission of this outbreak. So with data analysis, we would get all such information. And after getting all these information, our next step is the formulation of hypothesis. Why this outbreak has occurred? What is our hypothesis? We have to give a report of the why it happened. In this, we have to give about the possible source. What is the possible source of this outbreak? In case of food poisoning in a community after a wedding, is like the possible source may be the food served in the wedding. And depending upon the occurrence of sign and symptoms, depending upon the person distribution, we have to have an idea about the causative agent of the outbreak after we know about the source. Thirdly, we have to determine the possible mode of spread, why it has involved the population, why it did not uh, concise to a single population, why this outbreak has happened, what is the mode of spread, and of course the environmental factors that determine the occurrence of outbreak. So with all the data analysis, we have to frame our hypothesis on such points. After that, after formulating the hypothesis, our next step is the testing of hypothesis, whether our hypothesis is correct or not. How will we do that? In this case, we would be comparing the attack rates in various age groups. Since I already told you that this happens in case of infectious diseases, like in case of food poisoning, in case of suppose uh, measles, malaria, etc., just like infectious diseases. So we would be comparing the attack rate. How much of the population are susceptible? How much of them are involved with the sign and symptoms? How much of them are getting exposed to the disease agent? How many of them are getting sign and symptoms of that particular disease? So After knowing all these informations, we would be comparing the attack rate in various age groups. Okay, so after comparing the attack rates, we would be testing our hypothesis whether that hypothesis that we have framed is correct or not. Along with that, we would be evaluating the ecological factors responsible for the outbreaks. What are the environmental and ecological factors? Here, we would be dealing with the circumstances, the evidence, provide circumstantial evidences in the environment. And these are important because further transmission of disease do not occur. So ecological factors evaluation is important in case of outbreak investigation. Okay. Further, after doing the ecological factors evaluation, we would be investigating the population again, because after testing the hypothesis, if we find that our hypothesis is correct, we would be visiting the population and we would be finding the cases, and then we would be investigating those cases in details to obtain the additional information. Okay, it is done by medical examination and tests, and this time the medical examination and tests are not screening, rather it is diagnostic. Okay, and in this case, suppose a case uh, is there in a house, and in the same house few of the people are there who are not affected, so we will term them as controls, or rather healthy individuals who are not the cases. So we can study it in a case control manner, that why such and such population are affected and why the other group are not affected in the same household. Okay, so in this way our further investigation of the population is being done to know more about the disease or outbreak. And the final step comes out to be the report writing. After knowing all such information, till the ninth step, we will frame and write our report. In report writing, we must ensure that it is complete. Okay, and when I say complete, it must include such points. Number one, background. In background, we have to write about the uh, outbreak, that what kind of outbreak is it. Okay. Secondly, the historical data. In historical data, we have to mention that what are the cases that were frequently observed in the population before the outbreak. That means I am talking about the endemic nature. And why did you call this an outbreak? You have to justify your point with the historical data. Okay. Thirdly, the investigation methods. That what are the methods you have carried out in this investigation of an outbreak? You have to mention about the team members. Okay, how was it started, who are the team members and what are their qualifications. Fourthly, when and how the house to house survey was started, how the population at risk was calculated, was contacted, how the contact tracing was made. So all the steps that you did in calling it an outbreak and giving your points for it, you have to mention it in the investigation of an outbreak methods. Then you have to show a detailed data analysis method. After collecting all such data, how the data analysis was done and what was its interpretations, we have to mention it in our report. And after the interpretations, we have to give the recommendations for preventive and control measures of an outbreak. With this, you can say that your report is complete. Okay, I repeat again, in this slide, your report must be complete. You have to give the background details of the outbreak that has occurred. You have to verify and justify that why you have labeled it as an outbreak based on the historical data. You have to introduce your team members with their qualifications and how these team were selected, and what are the factors you have counted upon in house to house survey, what was the case definition, what were the sign and symptoms did you face, and how the contact tracing were done. Okay, and after obtaining these informations, how data analysis was done, what were its interpretations, and depending upon the interpretations, what were the recommendations for preventive and control measures of this outbreak. You have to write it down in a clear manner. That will complete the report writing. So that was all for in today's lecture. Thank you. And if you have any questions for me, uh, kindly comment below. So that was all for today. Uh, but let me discuss one more thing before I conclude. Is that uh, this often comes as a long question, the investigation of an outbreak in the exam. It may come to you in 10 mark or a 5 mark. Sometimes um, in a spotting of in a practical exam, it is given the different epidemic curves. When will you say it is point source, or when will you say it is propagated? So all such diagrammatic figures would be given, and then you will be asked about what type of epidemic curve is it. So do study. Thank you once again.